Hi guys, moving on to chapter 13, talking about another type of muscle. In this case here specifically cardiac muscle. So we're going to go over two types of cardiac muscle. Either one, both of them are outstandingly, if I can use that word, very, 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 very interesting. The cardiovascular system is also very, very interesting. It actually rivals the nervous system. I'm biased toward the nervous system, but cardiovascular system is also very challenging but gorgeous as an organ system. So it should be exciting to go over the entire organ system. Cardio means the heart, vascular means the blood vessel. But the cardiovascular system actually consists of three components: the heart, the blood vessels, and obviously blood, right? So going over each component individually, starting with chapter 13, focusing on cardiac function, meaning the heart itself. And when we talk about cardiac function, the heart has two components. It has an electrical component and it has a mechanical component. So when we say cardiac cycle, we're talking about the mechanical component of the heart. Okay, mechanical, MECHANICAL. So we have electrical and mechanical component of the heart, right? So cardiac function includes two components. So when we say cardiac functions, we are talking about the electrical component and the mechanical component of the heart. So what is also interesting is that electrical component dictates the rate of the heart. And what is amazing about this functionality of the heart is the heart does not require neural input, meaning the heart does not require decisions coming from the central nervous system. In other words, if you take the heart out of its host, the heart it beats. It beats until it runs out of nutrients and oxygen, but it still beats. And it not only it beats on its own pace. So that heart is independent from the nervous system. And that is unique. Well, not unique. Its own smooth muscle has the capability as well. But that's one of the most fundamental difference between cardiac muscle and skeletal muscle. Skeletal muscle requires neural input 24/7. Skeletal muscle absolutely requires the nervous system to come in and tells it what to do. As soon as that neural input is cut off, skeletal muscle dies. Cardiac muscle, on the other hand, it does not need that innovation. Don't get me wrong, the central nervous system does influence cardiac muscle function, but it is not an absolute requirement. Whereas in the skeletal muscle case, it is an absolute requirement, right? So for skeletal muscle to work normally, it needs central nervous system input. It needs that lower motor neuron to come in and synapse with, not synapse, but make a neuromuscular junction with the skeletal muscle fiber. So the skeletal muscle fiber can contract. You cut that off, the skeletal muscle fiber will die. In the case of the heart, it won't happen. The cardiac muscle fiber still contracts happily, even if the brain does not intervene. And then the cardiac muscle, the cardiac cycle, the mechanical component, this is the component where the heart is pumping blood, right? It's generating the pressure to basically push blood through the entire vascular bed, through the entire circulatory system, through the entire, blood vessels, meaning all the arterial vascular bed, through the capillary bed, back to the heart through the venous bed, right? Arteries, capillaries, veins. And then the amount of blood that's being ejected by the heart is called cardiac output. And we're going to go over factors influencing that cardiac output. How much blood is being ejected, pushed out by the heart, depends on specific factors. And we're going to see those factors during today's recordings. So with that said, let's begin the lecture, starting with the first slide. Oh, before I get to that, I'd like to mention something. When we talk about cardiac muscle, there are two types of cardiac muscle. There's the pacemaker cells and the actual contractile cell. Those are basically contracting. So both of them are two types of cardiac muscle cells. Okay. So the pacemaker cells are bona fide cardiac muscle cells. The difference is these pacemaker cells have lost the ability to contract, have lost the ability to generate cross-bridge formation, but have gained a very, very nice ability. And that ability is to spontaneously generate action potentials. So these pacemaker cells have lost one function, but have gained another function, right? So they've lost the ability to contract, but they have gained an ability to generate, to produce spontaneous action potentials. And these action potentials will dictate the rate of the heart, the rhythm of the heart. So pacemaker cells and cardiac contractile cells. So when we say in anatomy cardiomyocyte, we are referring to the cardiac contractile cells, right? The pacemaker cells in anatomy, we call them part of the conducting or conduction system, right? So for example, the AV node SA node, all of that is basically consistent of pacemaker cells. Whereas the myocardium contains the actual cardiac muscle fiber that generates cross bridge formation to contract the heart, to generate pressure to push blood out of the heart into the vascular bed. Now we are ready to start the lecture. This slide is a very important slide because it contains at least two new concepts. The first one being that when we talk about in neurons, when we discuss and we learn about the depolarization here, in neurons, the depolarization is carried by a massive influx of sodium cations into the neuron, right? We learn that. In the heart, there's that too. So there's the depolarization is also carried by sodium influx. But that's when we talk about the contractile cardiomyocyte. But when we talk about the pacemaker cells, this rapid depolarization is not mediated by a massive sodium influx into the cardiac pacemaker cells. Okay, we're going to talk about it. So that's the first concept that you're going to learn something new today. The second concept is the repolarization signal activates ion channels. We learn from neurons that when we talk about repolarization here, we are talking about opening a voltage sensitive potassium channel, the repolarization. But down here, the hyperpolarization is not about channel opening in neurons. It's still the same voltage sensitive potassium channel that remains open. But in this case, in the pacemaker potential, that repolarization, that hyperpolarization potential actually opens novel types of ionic fluxes, novel types of ion channels that we're going to talk about very shortly. So those are the two new concepts. Concept number one, depolarization is being carried by another type of ionic species, not by sodium, at least in the pacemaker cells. And in the hyperpolarization territory, the potential that's below more negative than threshold actually opens new types of ion channels. Okay. So with that introduction, let's begin. So as you can see here, in the pacemaker potential, which is this entire region, that's the pacemaker potential. That's a spontaneous potential that we talked about. The pacemaker cells have this capability of generating spontaneous depolarization. Okay, spontaneous, spontaneous, spontaneous. Action potential. Okay, which is called pacemaker potential. And this pacemaker potential actually has two parts to it. There's a first part here, and the second part here in yellow, right? So the two different parts mediated by two different ionic currents, or the current's influx is mediated by two different ionic species. Now, if you follow with me, this picture is from page 371, figure 1312. But what is important, I'm going to ask you to flip over on the other page, 372, and you can follow me with the table 13.1. That table is important to understand this picture here, the panel B of picture figure 13.12. Okay, so this is figure 13.12, but you need that table 13.1 to follow. So let's start with the first phase. Let's use another color. So let's talk about this phase here. And that is due to sodium influx and potassium efflux through a channel by the name of funny channel. I know. Okay. Funny channels, Funny channels. And the funny channels, they open in this area here, in the hyperpolarization territory, and they flux two ionic species. They allow sodium to get in, so sodium influx. They also allow potassium efflux, so they are permeant to two different ionic species. So there's simultaneous sodium getting in and potassium getting out of the pacemaker cell, right? Now, there's much more sodium getting in than potassium getting out. So there's a net depolarization, meaning going toward more positive membrane potential value, moving toward the threshold. Unfortunately, the threshold is sitting at minus 50, right? But the funny channel can get only to minus 55. Millivolts. So they will be short by 5 millivolts. They will fail to get to threshold potential, to fire that action potential, right? So the minus 55 is 5 millivolts short from the threshold. So this is the threshold right here. The threshold potential, the same concept, right? So for pacemaker cells, it's at minus 50. So if you get to minus 55, it's not enough. But that depolarization is enough to cause an opening of another type of ion channel, which is over here in the yellow bar. And this is due to a massive calcium influx into the pacemaker cell through another type of channel. Okay, this calcium influx is through what is called the T type calcium channels. So that depolarization caused by the Fundy channel opening is actually opening this T-type calcium channel. T stands for transient. They open only briefly, transient. Transient. Okay, stands for transient calcium channel. And the transient calcium type calcium channel opens enough calcium to get in to depolarize, so to bring that membrane depolarization minus 55, which is 5 short of threshold, bring it to minus 50 to get to threshold. So this T-type calcium channels allows the cell membrane, allow the plasma membrane, allow the phospholipid bilayer of the pacemaker cell its potential to get to minus 50 millivolts, so getting to threshold to fire this action potential. And this depolarization, when you get to threshold, you obtain that depolarization. And this depolarization it's also caused by a massive calcium influx. This depolarization, this is new, this is not from neurons. So this is depolarization is mediated by massive calcium influx. Okay, and this calcium influx is not through the same calcium channel. Now this is called the L type. L type calcium channel. Okay. This word L here stands for long lasting, meaning they stay open longer to allow that calcium fluxing in to depolarize the cell membrane, the plasma membrane, the phospholipid bilayer of these pacemaker cells. And that action potential happens spontaneously, and the repolarization now is similar to neurons. This is potassium channels open, allowing potassium efflux, and the membrane repolarizes. And when it hits down here, this is the resting membrane potential, RMP. and it's not stable. This resting membrane potential, resting potential here, RMP, resting membrane potential is unstable. The neurons, in neurons is stable, but in the pacemaker cell, it is unstable. So it fluctuates, so lacking steady resting potential. So as it gets down here, it hits the resting membrane potential, it activates funny channels. Funny channels open, they shut. In turn, the transient calcium channel open, they shut, they activate open the L-type calcium channel, mediating depolarization, they shut, open potassium channel, going back to repolarization, and the cycle starts again. Okay. So this is a fantastic mechanism. So spontaneously, these pacemaker cells, also known as autorhythmic cells, are capable of generating action potential on their own without the um, input from the central nervous system, without the intervention of the brain and or the spinal cord, right? So the heart itself has this capability of generating its own action potential. Which is not possible, by the way, for cardiac muscle. The cardiac muscle can generate action potential, but it requires neurotransmitter release from the lower motor neuron. So they require neural input, not cardiac muscle. Huh? So you need to learn that table 13.1 on page 372. So it allows you to follow this picture here, the panel B from figure 13.12 on page 371. So this again, one more time, this panel B here is from 13.12, figure 13.12 on page 371 of your Stanfield textbook. And this picture goes hand in hand with table 13.1 on page 372. So make sure that you learn that table and you understand this panel here. I'm not playing games with you. I'm telling you upfront, right? This concept will be assessed by both the lecture exam and the lab test. So make sure that you understand the concept. Now moving on to the same thing, but now we're going to focus on the cardiomyocyte, the actual cardiac muscle fiber that generates cross-bridge formation to generate tension, to contract the heart, allowing the heart to create pressure, to pump blood, to eject blood from the heart into the vascular bed. Okay? Now this picture here is from figure 13.13 on page 372. And it goes hand in hand with the table 13.2 on page 373. Okay, you learn that table as well because it allows you, it describes these different phases here. Okay, so let's go over this. There are 5 phases, not 4. The phase 0, 1, 2, 3, 4. So 1, 2, 3, 4, 5, right? So phase 0, phase 1, phase 2, phase 3, phase 4. So there are 5 phases in an actual action potential for a cardiac contractile cell. Cardiac contractile cell. Very similar to neuron, they have stable resting membrane potential. So stable RMP. It says resting potential here. It should have been resting membrane potential to be technically correct, right? Now, these different phase description, phase 0, phase 1, phase 2, phase 3, and phase 4 are described clearly by 5 short paragraphs that you need to go over and read that on page 372 and 373. So briefly, the phase 0 here is caused by a massive sodium influx. So this is very similar to neurons, okay? Phase 2, maintain the depolarization. This is due to calcium influx. And this calcium influx goes through the L long-lasting L-type calcium channels. In the case of cardiac contractile cells, there are 2 types of voltage, 2 types of potassium channels that we need to learn as well. So during phase 1, the potassium channel is called inward rectifier channels. 3 words, I'm going to spell it out for you. Inward, spell INWARD, inward. Second word, rectifier, 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 channels, right? CHANNELS. So inward, inward, rectifier, channels. And this is a type of potassium channel. And they are closed during phase 1. Okay, so this inward rectifier channels are also a voltage sensitive potassium channel. The book says voltage gated. No, it's voltage sensitive potassium channel. And specifically, they have a name here. They are called inward rectifier channels, and they are closed during phase 1. So basically, this part here, repolarization, is being delayed. Okay. And simultaneously to that, since there's no potassium efflux, so you're basically slowing down repolarization, and then you open another channel to allow potassium influx, so you're depolarizing the membrane. So you keep that depolarization, you extend it out, so it basically plateaus out instead of going down in the repolarization here, right? So the plateau is mediated by two components: the closure of the inward rectifier and the opening of the L-type calcium channel. Okay, so the plateau is mediated by the closure of the inward rectifiers and the opening of the L-type calcium channel. And the repolarization this is caused by an opening of another type of ion channel called delayed rectifier channel. Again, delayed rectifier channels. So the word delayed, D-E-L-A-Y-E-D, delayed. Second word rectifier, the same word as before, R-E-C-T-I-F-I-E-R, and channels. So here's the delayed rectifier channels. They open to allow potassium efflux to repolarize phase 3. And also during this repolarization here, the inward rectifier channels are also open. So both of them mediate the potassium efflux. So potassium is leaving the muscle, the cardiac muscle cell, to repolarize its cell membrane. Okay. And then going back to the steady, stable phase 4, which is the resting membrane potential. So this is important, and not only also for the cardiac muscle, contractile cardiac muscle, the resting membrane potential is at minus 90 millivolts. This is the RMP, resting membrane potential. Because for the pacemaker cells, the resting membrane potential is minus 70, not minus 90, okay? The minus 90, that's for the cardiac contractile cell, the cardiac muscle cell. The pacemaker cell, the resting membrane potential sits at minus 70, not minus 90. So there are very, very different behavior, membrane potential behavior for the two types of cardiac muscle cell. The autorhythmic cell / pacemaker cells versus cardiac contractile cell / cardiomyocyte. And we talk about this also, the importance of the gap junctions. So what is amazing is the action potential travels between neighboring cells through gap junctions. So technically speaking, all you have to do is you innervate one single contractile cell. Right? I'm going to cross out the X to emphasize. All you need is to activate one single cardiac contractile cell. And you depolarize that cell, and the cell, the action potential travels through gap junctions to depolarize the neighbor. So you need to activate only one single cardiomyocyte, and the entire heart gets contracted, gets excited. In the case of the skeletal muscle, every single skeletal muscle fiber, every single skeletal muscle cell requires a neuromuscular junction with the lower motor neuron axon terminal. In the case of the cardiac muscle fiber, there's no such requirement, right? The pacemaker cells comes in, excite one single cardiomyocyte, the entire heart gets excited through gap junctions. Again, the minus 70 millivolts, that's for the resting membrane potential for pacemaker cells, autorhythmic cells, whereas the minus 90 millivolts, that's the resting membrane potential for cardiac contractile cells or cardiomyocytes. Now, moving on to the mechanical component of the heart. So when we say cardiac cycle, we are now officially out of the electrical component, right? So the EKG, that's the electrical component. Cardiac muscle, cardiac cycle, that's the mechanical component. So when we say cardiac cycle, we are talking about the mechanical component of the heart. When we say mechanical component, we're talking about the pumping component of the heart. Right? And when we say the electrical component of the heart, we are talking about the rhythm of the heart. Rhythm, R-H-Y-T-H-M, rhythm. Pumping, P-U-M-P-I-N-G. Now, in the clinical setting, there's no way for us to monitor the mechanical component of the heart non-invasively. So there's a cheaper, quicker, dirtier way, if you will, to follow the mechanical component of the heart indirectly by following the electrical component of the heart, the EKG. EKG stands for electrocardiogram. The K here stands for cardiogram, so you can say EKG, or you can replace the K for C or ECG, right? So this K here is replaceable with the C because that K cardiogram is in German. So EKG and ECG means the same thing. It's the reflection of the electrical activity of the entire heart. And since they are electrical signals, they are being propagated to the surface of the body, which can be picked up by electrodes placed on a specific location on the body. And we get this unique characteristic profile that we call an EKG, showing the electrical component of our heart. And you see here, this is one single EKG. Okay, this is 1 EKG equals 1 heartbeat. So here showing you 3 EKGs, 3 heartbeats. So if your heart beats like 70 times per minute, within that minute, within that 60 seconds, you will have 60s of these EKGs. Okay. So 1 EKG equals 1 heartbeat. And this EKG is electrical activity for the entire heart. And this is for coming from 1 cardiomyocyte. So this is 1 cell. This is for the entire organ. So this is 1 cell again, an action potential within 1 single cell. This is a summation of all potentials for the entire heart. Okay, so this is cellular level, this is organ level. So membrane potential for 1 single cardiomyocyte. Let me write it down. So this here is 1 AP action potential from 1 cell, right? This cell here is not the pacemaker cell. This cell is the cardiomyocyte. This cell is the contractile cell, right? Whereas here, this is 1 EKG for the entire heart, okay? Let's go over this. The P this part here, which is this one, it's called P deflection. And that P deflection, this is caused by atrial depolarization. So both atria are being depolarized. Both atria are being activated, right? So that's the electrical component of the heart. The depolarization, that's the electrical component or the conduction system. And that's important. You're not looking at the mechanical component here. The mechanical component is not shown on an EKG. However, you can infer on the EKG. So halfway through the P deflection, you will have atrial systole. Atrial, A-T-R-I-A-L. Systole, S-Y-S-T-O-L-E. Systole means contraction. So that's the word right there. Atrial systole, that's the mechanical component. This is when both atria are contracting. And they can only contract after they're being depolarized. So that's why you have to wait until around midway through the P deflection. Your atria, both of them will go into contraction, will go into systole, okay? So from the EKG, you can infer the cardiac cycle, okay? So that's important. EKG allows you to infer on the cardiac. So infer, I-N-F-E-R. Infer, I-N-F-E-R. So looking at the EKG, we can make an inference to the cardiac cycle. Okay, we can deduce the mechanical component of the heart. So following the P deflection, you have another massive signal. It's called the QRS complex, which is, this part here. Okay. So Q, R, S. Look at the better drawing. This point, and that point. So you go up, and you go straight down. So this is the Q. This is the R. And this is the S. And we call that Q, R, S complex. And the QRS complex includes two events. Includes the atrial repolarization, right? So both atria are now repolarized. They go back to rest. Simultaneously, you have ventricular depolarization. So now the ventricles, both of them are being depolarized. So both of these events are shown by the QRS complex. But the QRS complex are reflecting more the ventricular depolarization. It's masking the signal coming from the atrial repolarization, which is a weaker electrical signal. So it's basically being buried by the larger ventricular depolarization that we call QRS complex. So the QRS complex actually is being, caused by two events. One larger ventricular depolarization and one smaller atrial repolarization. Okay. So that during that QRS complex, we can infer the atria are relaxed and the ventricles are contracting. So following atrial repolarization, the atria are in diastole. So atrial diastole. Diastole means relaxation. The muscles are relaxed, right? The atria are relaxed. Diastole, D-I-A-S-T-O-L-E. Okay, so atrial, A-T-R-I-A-L. So both atria are in diastole, meaning both atria are now relaxed. Whereas the ventricles, right after depolarization, they will be in ventricular systole, VS, right? Ventricular, V for ventricular, S for systole. Again, systole, S-Y-S-T-O-L-E. The ventricles are now contracting. And after ventricular systole or depolarization, the ventricles now go into repolarization, and that's shown by, reflected by the T deflection. Okay, so this is called T deflection. Yeah. Deflection again, D-E-F-L-E-C-T-I-O-N, T deflection. And this is reflecting ventricular repolarization. So both ventricles are now repolarized. They're going back to rest. And following the ventricular repolarization, you will have ventricular diastole, right? V for ventricular diastole, D-I-A-S-T-O-L-E. The ventricles are now relaxing. They're not pumping, okay? So those are the different deflections on an EKG. Now, let's take it to another level. This is clinical implication. This is how we use in the clinical setting. There are important intervals or segments. So the QT interval, that's the time, the duration of your ventricular systole. This is the time, the duration during which your ventricles are contracting. So the QT interval are very important right here. This is the amount of time during which both ventricles are contracting. So that's the QT interval. The TQ segment, which is this one here, this is when both ventricles are relaxed. They're not contracting. And the RR, that's the time between each heartbeat. So let's say if you have 70 beats per minute, HR will be around 0.8 second. And the PQ, which is not shown on here, it's important to write that down. The PQ is the duration exactly when the time when both atria are in systole, meaning both atria are contracting. Okay, so PQ, this is the duration for atrial systole. QT, ventricular systole. TQ, ventricular diastole. And then RR, that's for between heartbeats. These are the different phases of the cardiac cycle. Two things that are important. Isovolumetric contraction and isovolumetric relaxation. Both of these, all cardiac valves are closed. Okay, again, all four cardiac valves are closed. The two atrioventricular, the right atrioventricular, the left atrioventricular, and the two semilunar, the aortic semilunar, and the pulmonary aortic semilunar valve are closed. The difference between the two, even though all four valves are closed, are caused by different factors. When we say isovolumetric contraction, this word here, the heart, the ventricles are starting to squeeze, the ventricles are contracting, starting to generate pressure. But that pressure is strong enough to close the right and left atrioventricular valve, meaning the valve between the ventricles and the atria. The pressure is also building up, but it's not strong enough to blow open the semilunar valves yet. So both the aortic semilunar valves and the pulmonary semilunar valves are still closed, right? So during IVM contractions, the ventricular pressure, the ventricles are starting to contract. The pressure is being building up, and that building up pressure shut close the AV valves, but that pressure is not strong enough yet to open the semilunar valves. That's why all the four valves are closed. And when we have the isovolumetric relaxation, isovolumetric relaxation is when the ventricles start to relax. And when the ventricle starts to relax, the pressure inside the ventricles are dropping, but they're still too high to open the AV valves. Okay? And the inside the pressure is also too high. It's basically less than the blood pressure in the aorta, ascending aorta, and the pulmonary trunk. So blood flushes back toward the ventricles to shut close the semilunar valves. So all four valves here also are closed during isovolumetric relaxation, meaning both ventricles are relaxed, but the pressure inside of them is still too high to allow the AV valves to open. And the pressure now is less than the blood pressure in the ascending aorta and the pulmonary trunk. So blood flushes back, try to get into the ventricles. That's why they force the semilunar valve to shut close, preventing backflow. So that's during isovolumetric relaxation. So both cases, IVM contraction and IVM relaxation, this is the only time when all four valves are closed within the heart. And then the meaning of the lub and the dub. That's the heart sounds. The lub is basically low pitched, And the dub is basically high-pitched, louder sound. The lub is due to the closure of the AV valves. So this happens during here, the heart sound. That's for the lub. That's the closure of the AV valves. And for the dub, the louder sound happens here during phase 4. And that's due to the closure of the semi-lunar valve. Now, there's a misconception from students, most of us, not all of you. They think that the fact that when the AV valves close and the semilunar valve close, that's a mechanical, when they shut closed, they give rise to a sound. And that's not true. That's incorrect. When the valves close, they don't make any noise. They don't make any sounds. The sound that we hear is due to blood turbulence. So when blood comes back and they strike on the valves, and basically the turbulence in the liquid, in that blood, it's what makes, we pick that as a sound, right? So the low pitched, called lub, that's due to blood turbulence caused by the closure of the AV valves. The dub, the louder sound, is caused by blood turbulence caused by the closure of the semi-lunar valves. Now, factors that affect the heart, the cardiac output. So when there's a equation that will come back. CO stands for cardiac output equals HR, heart rate, multiplied by stroke volume, right? So cardiac output is the amount of blood being ejected from the heart. And that amount of blood being ejected from the heart depends on 2 factors. The first factor is the heart rate, how fast the heart beats, and stroke volume, how strongly the heart contracts. So as you can see, right, the heart rate is the electrical component of the heart, whereas the stroke volume is the mechanical component of the heart. So MC, mechanical component. HR, electrical component. So we've been talking about it. So electrical component, mechanical component. So the 2 components of the heart come together as a team to dictate how much blood the heart can push out. And that volume of blood that the heart is ejecting out of itself, we call it cardiac output. Cardiac, C-A-R-D-I-A-C, output, O-U-T-P-U-T, right? Output. So these are the 2 factors controlling, affecting, dictating CO. So we're going to talk about heart rate first. So factors affecting the heart rate. So if you increase the heart rate, you will increase cardiac output. If you increase stroke volume, you will also increase cardiac output. Or you increase both, you will also increase cardiac output. So the amount of blood, the volume of blood that's being pumped out of the heart depends on the heart's electrical component and also depends on the heart's mechanical component. So let's focus on the first factor, right? The heart rate. So what are the factors that can affect the heart rate? So the heart rate It's controlled by extrinsic factor. Extrinsic factor means factors coming outside from the cardiovascular system, right? Like the nervous system. So extrinsic means outside. Factors that are not belonging to the cardiovascular system. So let's begin here. The first one, the sympathetic division of the autonomic nervous system, ANS. It comes back, guys. Okay, we've seen this. So in this case here, the sympathetic division of ANS, it releases either norepinephrine or epinephrine. So let's focus on norepinephrine. And norepinephrine is released as a is acting as a neurotransmitter. Okay. And because it is released by the postganglionic neuron. Again, one more time, norepinephrine is released by the postganglionic neuron of the sympathetic division of the autonomic nervous system into a neuromuscular junction. And the neurotransmitter is called norepinephrine. The other one, just change color. Epinephrine. It is also released by the autonomic nervous system, but here is released as a hormone. Is released into the bloodstream by the adrenal medulla, specifically by the chromaffin cells within the adrenal medulla. So the chromaffin cells are modified ganglion, or modified ganglia, right? So they release a chemical messenger, in this case epinephrine, into the bloodstream. So epinephrine works as a hormone. Oops, I misspelled the word. Hormone. Okay. Neurotransmitter, neurotransmitter. Hormone, hormone. Yeah. Now, once norepinephrine or epinephrine shows up, this is the cell membrane of a pacemaker cell. Okay, one more time. This is the cell membrane, the plasma membrane, the phospholipid bilayer of the pacemaker cell. Not the cardiac contractile cell, because we're dealing with heart rate here, guys. Okay, so we're talking about the electrical component of the heart. Pacemaker cells, autorhythmic cells. So it doesn't matter at this case whether it's norepinephrine or epinephrine. Everything else is the same. Norepinephrine, epinephrine bind to the same receptor, the G protein coupled receptor called beta 1. The beta 1 G protein coupled receptor recognizes and binds to norepinephrine as a neurotransmitter or epinephrine as a hormone. Doesn't matter, like I said. In this case, they're both recognized as a chemical messenger, binds to beta 1. The receptor activates the G protein called GS, more specifically the alpha subunit of the G protein recognizes and binds and activates the adenylate cyclase, membrane-bound enzyme. The adenylate cyclase converts ATP into an important second messenger, cyclic AMP, and cyclic AMP activates protein kinase, which is an enzyme that is capable of adding phosphate group onto other target. So the protein kinase activates the funny channels, simultaneously activates the transient type calcium channel, allowing more sodium, more calcium to come in to depolarize the membrane. Remember, this is a pacemaker cells. So in other words, allowing the pacemaker cell to generate more action potential, right, per unit time. So what the outcome here is the heart rate increases because the number of spontaneous action potential produced per unit time increases. Now the opposite, the parasympathetic. The parasympathetic, we learned this also. This is also extrinsic, right? And in this case is the acetylcholine, ACH, as a neurotransmitter. This is also released by the post-ganglionic neurons from the parasympathetic division of the autonomic nervous system. And this ACH molecule, uppercase A, uppercase C, lowercase h, acetylcholine, it binds to its receptor called muscarinic cholinergic receptor. This is not an ion channel. The nicotinic cholinergic receptor is an ion channel. The muscarinic cholinergic receptor is a G protein coupled receptor, right? So this muscarinic cholinergic receptor binds to the ACH as a neurotransmitter. It activates the G protein. Again, the alpha subunit and the alpha subunit. This G protein opens a potassium channel. Potassium is leaving. It hyperpolarizes the membrane of the pacemaker cell. Here, shuts the T calcium channel, so there's no depolarization. As you can see, the membrane is further going away from threshold. So it decreases the amount of spontaneous action potential being generated, leading to the heart rate being decreased due to the number of spontaneous action potential being produced per unit time drops, because the membrane potential, the potential across the cell membrane of the pacemaker cell goes further away from threshold due to this hyperpolarization signal here, caused by the opening of potassium channel, letting potassium effluxing, leaving the cell, right? Causing a massive net loss of positive charge. So the inner leaflet here becomes even more negative, going further away from threshold. So preventing the frequency of spontaneous action potential drops, meaning the number of spontaneous action potential being formed decreases per unit time. And that's due to the parasympathetic division. So the sympathetic increases the heart rate, the parasympathetic decreases the heart rate. And this is, we talk about this, the hormonal control, right? So epinephrine released from the adrenal medullae. Just ignore the, we're running out of time here. Ignore this. Just focus on epinephrine. Okay, so it's the same mechanism, nothing changed as before. So epinephrine and norepinephrine is the same thing. So what you see here is at rest is the gray curve. You have two action potentials at rest. The green one are sympathetic, so now you have three action potential. And parasympathetic is the blue, just one action potential. Right? So you increase the heart rate with sympathetic, you decrease the heart rate with parasympathetic. Okay. So let's do that. You have one, so two APs at rest. You have three APs under sympathetic. You're down to one AP under parasympathetic. So that's what it means. So we understood the molecular level, the beauty, molecular mechanism explaining this observation, right? So now we understand how the parasympathetic slows down our heart rate. We also understand now how the sympathetic increases our heart rate. Okay? We're just applying what we've learned. There's nothing new here in terms of these receptors and the chemical messengers, either hormone or neurotransmitter. So that was the electrical component. Now we're moving on to the mechanical component, the stroke volume. Now this is important, right? For the mechanical component, the parasympathetic division cannot influence how strongly or how weakly the heart contracts. The parasympathetic can only influence the electrical component, can only slows down the rhythm of the heart. But the parasympathetic has no say in the mechanical component of the heart. Only sympathetic division can increase the strength of contraction of our heart. So only the sympathetic can increase the stroke volume. The parasympathetic has no say whatsoever in the stroke volume. Okay, so here, this is what it means now. So either norepinephrine or epinephrine, same thing, norepinephrine acting as a neurotransmitter, epinephrine acts as a hormone. Binding to beta 1, same thing happens. G protein gets activated, the alpha subunit activates the adenyl cyclase, and cyclase converts ATP to a cyclic AMP. Cyclic AMP activates the protein kinase. So far, it's the same, right? Up to this point. And here is when it deviates. The protein kinase phosphorylates the L-type calcium channel. Because here we are no longer in the pacemaker cell. We are now in the contractile cell, right? So this is the cardiomyocyte, right? So this is the cardiac muscle cell that, forms a cross bridge formation. So protein kinase activates and opens the L-type channel, prolonging the depolarization on the contractile action potential, more calcium gets in. So this calcium influx promotes cross bridge formation, the same mechanism we learned from skeletal muscle. Protein kinase also activates and opens a calcium channel on the membrane of the sarcoplasmic reticulum. Induce them to open, calcium leaves the sarcoplasmic reticulum into the sarcoplasm. So you have this calcium, you have this calcium. They both promote cross bridge formation. Okay. But protein kinase also affects the ATPase. So protein kinase increases the activity of the ATPase on the myosin heads. So it induces the myosin actin cycling. It increases the number of cycles per second. So it optimizes how many times myosin disengages from actin and then binds another actin. So we know that in skeletal muscle, it happens around 5 times per second. So it can basically increase that number of cross-bridge formation, the cycling between actin and myosin in the cardiac muscle, the contractile cardiac muscle cell, the cardiomyocyte in the same way. So protein kinase can make the cross-bridge cycle more efficient. So the first three steps, step 1, step 2, and step 3, they all promote contraction. Step 4 is unique. Step 4 is different. Step 4 is the opposite. Step 4 promotes sequestration of calcium. So step 4 promotes sarcoplasmic reticulum to take back calcium to promote relaxation. So when there's no more calcium, there's no more cross-bridge formation, right? So the protein kinase here actually activates the calcium ATPase. So this ATPase it's a pump that consumes ATP to pump in calcium. This is a uniporter. It only pumps in calcium. It takes calcium from the sarcoplasm, meaning the cytoplasm of the cardiac muscle fiber, and shoots it back into the sarcoplasmic reticulum, right? At the expense of ATP. So protein kinase phosphorylate this calcium ATPase, activates it, and then that calcium ATPase pumps calcium from the sarcoplasm into the sarcoplasmic reticulum, thereby ending cross-bridge formation, allowing cardiac muscle to relax. Same thing here, hormonal, same mechanism, nothing changes. In this case, the epinephrine acting as a chemical messenger. Yes, again, due to time, we are not going to talk about this. We're going to focus only on epinephrine. And learn this. So you should be able to understand, answer this. Why is epinephrine acting as a hormone and why norepinephrine acting as a neurotransmitter? We addressed this earlier in a few slides earlier, so you should be able to answer that question. Another thing of affecting stroke volume is what is called the end diastolic volume, and this is important. We talked about this briefly. Cardiac muscle at rest, they're not at the optimal length. So when the heart is filled with blood, incoming blood into the heart will stretch the heart, will expand the heart. So by the expansion, it stretches out the heart. And by stretching it out, it induces the sarcomere into a optimal position. So when the heart contracts, it allows the heart to generate much more tension. So this is the stretch here. The end-diastolic volume, the more blood comes in, you see that number one, that's the stretching out the wall of the heart. And that stretching out, you see here, the thin filaments are moving away from the thin line. This is too close, right? So that stretching, when the heart contracts, generates much more tension. So the stroke volume, this is much more volume increase due to that stretch. So this here, that's due to the stretching of the wall of the heart. The stretching lengthens out the sarcomere, right? And by doing that, it allows the sarcomere to produce much more. There's much more space to travel for cross bridge formation, allowing this is more tension generated. So more tension means more forceful contraction, more blood coming out of the heart. Stroke volume means blood coming out of the heart has been increased. Okay. So the amount of blood that comes back to fill the heart, we call it end diastolic volume. Okay, so that EDV is determined by EDP, so end diastolic pressure, and that end diastolic pressure, also known preload, affected by how much time the heart has to fill blood, depends on HR, heart rate. So the slower the heart rate, the more time the ventricles have time to fill blood. So the higher the EDV. Second factor, atrial pressure, depends on VR here, means venous return, how much blood comes back to the heart. So the more venous return, the more blood comes back, the stronger the stretch. The amount of stretching increases, thereby allowing more tension to be generated. The heart generates much more pressure, pushing out more blood. So stroke volume will increase. Okay. End diastolic volume means the volume that sits in the heart at the end of diastole. By the time when the ventricles are end of their relaxation, they have full amount of blood inside of their chambers, both the right and the left ventricle. We call that end diastolic volume, meaning the volume of blood inside each ventricle at the end of ventricular diastole, just before the ventricles contract. And lastly, afterload. This is not important under healthy conditions. It affects only individuals under pathological conditions. So the afterload determined by aortic pressure during ejection period. So what it means is the afterload is the amount of pressure that the heart needs to overcome before blood can be pumped into the aorta and also into the pulmonary trunk. Okay. So AL stands for afterload, back pressure exerted by arterial blood. Ventricles must overcome this pressure for B ejection, meaning blood ejection. So the aorta is 80 millimercury. For the pulmonary, it's only 8. So the blood pressure value is, we say 120 over 80, right? Millimercury. Millimercury. The top number is referred to as the systolic pressure. The blood pressure when the ventricles are contracting. The bottom number is called diastolic pressure. The pressure when the ventricles are relaxing. And the second number is the silent killer. We pay attention to the top number a lot in the clinical setting, but the true number, the true indicator of good long-term prognosis is the bottom number. The reason is the higher the number, the bottom number, the more pressure needs to be generated by the left ventricle, right? So let's say you have 80 millimercury. So in other words, the left ventricle needs to generate 81 millimercury before it opens the aortic semilunar valve. But instead of 80, the patient has now 95. So in other words, the left ventricle needs to generate more than 95 millimercury each time to blow open the aortic semilunar valve. So it puts more stress on the heart every time the heart contracts. So it puts more stress on the heart to be able to eject that blood out into the aorta. So that is a more dangerous number, the diastolic pressure, the lower number versus the top number, systolic pressure. Okay. And MAP, we're going to learn on the next recording. MAP is the driving force to push blood throughout the vascular bed, throughout the circulatory system. Mean arterial pressure. That's the driving force. That's the pressure gradient pushing blood forward, allowing blood to leave the heart, go throughout the systemic vascular bed, and come back to the heart. Okay. So that map we're going to learn on the next recording when we talk about the vascular bed. So on this note, I'm going to end the lecture today, and then I will see you during the next recording. And in the meantime, stay safe out there, be healthy, study well. Take care.